Hello.
Hello.
Now audible, madam.
Yes, sir, you're audible, but sometimes your voice is lagging.
Uh, so might be some net issue, I think.
Yes, sir.
So shall we continue now?
Hello.
Shall we continue, madam? Now? Hello.
Your voice is not properly audible. Hello, sir. Hello.
Just a moment. Money.
Yes, sir.
Just a moment. Just. Hello. Good morning, madam.
Good morning, sir.
So I think now it is okay, na?
Yeah, now it is proper.
Okay, madam. Shall we proceed now?
Yes, sir.
Please.
My screen is visible, sir.
Yeah. It is visible. So you can speak in front of camera now, madam.
Sorry, sir.
You can speak in front of camera, I think. You are not visible.
Oh, okay. I'm not visible. Mm, Hello.
Yeah, please, madam. Please
Can
proceed.
we? I'll proceed. Okay.
Yeah, please proceed.
Hello, everyone. Welcome to today's class at Tawhid Foundation for Allied Healthcare Science, Bangalore. In today's class, we'll be discussing the one second, sir. My screen is not moving. In today's class, we'll be discussing the ultrastructure of the bacterial cell for first year BSc MLT. Let's begin with the introduction. First, what exactly are bacteria? Bacteria are a type of biological cell that is prokaryotic and unicellular. Due to the lack of a membrane bound nucleus, these are simpler than other types of living organisms. Although only some of them can be seen by naked eyes, while the rest are microscopic, they display a wide range of shapes, size, and structures. Importance of studying bacterial cell structure. Understanding the structure of bacterial cells provides insight into how these microscopic, single celled organisms carry out vital functions to survive, multiply, and adapt to their surroundings. Bacteria are prokaryotes, meaning they lack a defined nucleus and membrane bound organelles. The cellular architecture is relatively simple, yet it contains all the necessary components to support essential activities like metabolism, growth, and reproduction. These organisms primarily reproduce by binary fission, a form of asexual division, and exhibit a wide variety of shapes and organizational patterns. Prokaryotic versus eukaryotic cells, brief comparisons. First, what is prokaryotic cells and eukaryotic cells? Prokaryotic cells are simple, primitive cells without a true nucleus or membrane bound organelles, for example, bacteria and archaea. Eukaryotic cells are complex cells with a true nucleus and a membrane bound organelles, for example, plant cell, animal cell, fungi, and protist. For prokaryotic cells, it lacks a true nucleus. The DNA is located in a region called the nucleoid, which is not enclosed by a membrane. Eukaryotic cells have a defined nucleus enclosed by a nuclear membrane, which protects the genetic material and regulates its interaction with the cytoplasm. Membrane enclosed organelles for prokaryotic cells do not have membrane bound organelles. Structures like mitochondria, endoplasmic reticulum, and Golgi apparatus are absent. For eukaryotic cells contains numerous membrane-bound organelles, each with specialized function, facilitating compartmentalization and greater cellular complexities. Chromosomes for prokaryotic cells possesses a single circular chromosome without histone proteins. For eukaryotic cells, have chromosomes arranged in pairs that are linear and associated with histone proteins, allowing complex DNA packaging and regulation. Cytoplasmic streaming for prokaryotic cells, no cytoplasmic streaming. The cytoplasm is relatively static. For eukaryotic cells, exhibit cytoplasmic streaming, a movement of cytosol and organelles that helps distribute nutrients and molecules within the cell. Cell division without mitosis for prokaryotic cells divide by binary fission, a simpler process without mitosis. For eukaryotic cells, divide by mitosis or meiosis, sophisticated processes ensuring accurate chromosome segregation. Simple flagella for prokaryotic cells composed of a single protein filament used mainly for locomotion. For eukaryotic cells have complex flagella and cilia with intricate structures that enable more versatile movement. Smaller ribosomes for prokaryotes contain smaller 70S ribosomes which translate messenger RNA into proteins. For eukaryotes, contain larger 80S ribosomes for protein synthesis in the cytoplasm. Mitochondria and chloroplast contain 70S ribosomes. Simple cytoskeleton for prokaryotic cells, helping in maintaining cell shape but less complex. For eukaryotic cell, complex cytoskeleton made of microtubules, microfilaments, and intermediate filaments, providing support, shape, and aiding intracellular transport. No cellulose in cell wall. For prokaryotic cells, most have cell wall made of peptidoglycan, which is absent in archaea. For eukaryotic cells, plant and fungal cells have cell wall made of cellulose and chitin, respectively. Animal cells lack cell wall. No histone protein. For prokaryotic cell, DNA is not associated with histone proteins, whereas in eukaryotic cells, DNA is wrapped around histone proteins, which help in organizing and regulating gene expression. So, general features of bacterial cells: unicellular and prokaryotic. Bacteria consist of a single cell that is prokaryotic, meaning they lack a true nucleus and membrane bound organelles. The genetic material floats freely in the cytoplasm. Second is lack membrane bound organelles. Unlike eukaryotic cell, bacteria do not have structures such as mitochondria, endoplasmic reticulum, or Golgi apparatus inside their cell. Reproduce by binary fission. Bacteria multiply asexually through a simple process called binary fission, where one cell divides into two identical daughter cells. Size ranges. Bacterial cell generally ranges in size from 0.2 to 2 micrometers in diameter, which makes them much smaller than most eukaryotic cells and visible only under a microscope. Bacterial cell structure overview. A bacterial cell is a unicellular prokaryotic organism characterized by several key structural components that together supports its survival, growth, and reproduction. So, major components contain cell wall, plasma membrane, cytoplasm, nucleoid, ribosomes, flagella, villi, and capsules. First, what is cell wall? A rigid outer layer made primarily of peptidoglycan. It provides shape, protection, and prevents the cell from bursting in hypertonic environments, differentiates bacteria into gram positive and gram negative. Plasma membrane, a phospholipid bilayer beneath the cell wall. It controls selective permeability, allowing nutrient in and waste out. It is a site of important metabolic activities like respiration and photosynthesis in some bacteria. Third is cytoplasm. Cytoplasm is a gel-like substance inside the plasma membrane. It contains enzymes, nutrients, ribosomes, and other molecules. Fourth is nucleoid. Nucleoid is a region within the cytoplasm where the single circular DNA chromosome is located. It is not bound by a membrane and act as the genetic control center of the cell. Fifth, ribosomes. Ribosomes is smaller than eukaryotic ribosome termed 70S and is responsible for protein synthesis. Flagella, it is a long whip-like structures used for motility, that is movement, helps bacteria move toward favorable environments or away from harmful stimuli. Then villi. Villi and fimbriae. It is a hair-like projections on the cell surface. Fimbriae assist in adhesion to surfaces and villi helps in bacterial conjugation, that is DNA transfer between cells. Last is capsules, a protective outer coating outside the cell wall. It helps in adhesion to surfaces and evasion of the host immune system. So this is a bacterial cell diagram. I'll explain each term. First, what is flagellum? This diagram illustrates the ultrastructure of a typical bacterial cell and its key component. So we'll start with flagellum. Flagellum are long whip-like appendages used for bacterial motility. It enables the bacterium to move toward nutrients or away from harmful substances. Pilus or pili, it is a hair-like projection shorter than flagella, facilitate attachments to surfaces and play a role in conjugation, transfer of genetic material between bacteria. Slime layer, it is a loosely attached gelatinous layer exterior to the cell wall, provides protection against drying and helping in adherence to surfaces. Cell wall, it is a rigid structure surrounding the plasma membrane, provides shape, mechanical support, and protection from osmotic pressure. It is composed of peptidoglycan in bacteria. Then cytoplasmic membrane. It is a phospholipid bilayer beneath the cell wall. It regulates entry and exit of substances and houses enzymes for metabolic activities. Then chromatophores vesicle. It is a membrane-bound vesicle containing pigments or enzyme often involved in photosynthesis or respiration in certain bacteria. Then ribosomes. Ribosomes are small particles scattered throughout the cytoplasm. It is a site of protein synthesis, which is 70S type in bacteria. Then mesosome. Mesosomes are nothing but infoldings of the plasma membrane. Proposed roles include cellular respiration, DNA replication, and distribution during cell division. Then RNA. RNA is messenger and ribosomal RNA involved in protein synthesis. Then nucleoid. Nucleoid is irregularly shaped region containing the bacterial chromosomes, which is circular DNA, and it is not enclosed by a membrane, the genetic control center of the cell. Gas vacuole. Gas vacuole is fill structures that provide buoyancy, helping in aquatic bacteria to position themselves in water columns. Polybeta hydrobutyric acid. It is a granule storing energy and carbon reserves in the cell. Then thylakoids, lamellae. Thylakoids are membranous structures involved in photosynthesis in photosynthetic bacteria, which is similar to chloroplast membrane in plants. Polyribosomes. It's a group of ribosomes bound to a single messenger RNA molecule synthesizing multiple copies of a protein simultaneously. Plasmid, it is a small circularly DNA molecules independent of chromosomal DNA. It carries genes that may confer advantages such as antibiotic resistance. Lastly, metachromatin granules or volatile granules. It stores granules of inorganic phosphate and serves as energy reserves or phosphate source. This is also bacterial cell diagram. Here Plasmid is nothing but small circular extrachromosomal DNA molecules found in some bacteria. It carries some additional genes that can provide advantages such as antibiotic resistance. Flagella, it is a long whip-like appendages that extend from the cell surface, responsible for bacterial movement, allowing the bacterium to swim through liquids. Then capsule, it is a thick protective outer layer surrounding the cell wall, helps in protecting bacteria from desiccation and host immune attacks, and aids in adherence to surfaces. Chromosome DNA, the main genetic material of the bacterium, typically a single circular chromosomes located in the nucleoid region. It contains all the essential gene for the cell's function. Ribosomes, it is a small particle scattered throughout the cytoplasm, site of protein synthesis where amino acids are assembled in proteins. Cell membrane, that is plasma membrane, a phospholipid bilayer beneath the cell wall, regulates the movement of substances into and out of the cell, maintaining homeostatistics. Cytoplasm, it is a gel-like substance inside the cell membrane containing enzyme, nutrients, and cellular compounds. Cell wall, a rigid protective layer outside the cell membrane, provides shape, mechanical support, and prevents the cell from bursting due to osmotic pressure. Now, what is cell wall? Composition of peptidoglycan. The bacterial cell wall is primarily composed of peptidoglycan, a unique polymer that forms a mesh-like structures. Peptidoglycan consists of long chain of sugars cross-linked by short peptides. This combination provides both strength and flexibility. Provides shape and protection. The rigid peptidoglycan layer maintains the characteristic shapes of the bacterium, whether spherical, rod shaped, spiral, or other forms. The cell wall acts as a protective barrier, preventing the cell from bursting, especially in hypotonic environments where water would tend to enter the cell. Gram positive versus gram negative differences. So gram-positive bacteria have a thick peptidoglycan layer up to 20 to 80 nanometer thick. This thick layer retains crystal violet stain used in gram staining, making this bacteria appear purple under microscope. It contains teichoic acid in the peptidoglycan, which play roles in cell wall maintenance and ion regulation. Gram-negative bacteria have a thin peptidoglycan layer around 2 to 7 nanometer situated between two membranes, the inner plasma membrane and an outer membrane. The outer membrane contains lipopolysaccharides, LPS, which can act as endotoxin and contribute to pathogenicity. It do not retain crystal violet stain. Instead, they take up the counter stain, which is safranin, and appear pink or red in color. The outer membrane provides additional protection against certain antibiotics. So now characteristics of gram-positive and gram-negative bacteria. Outer phospholipid layer. For gram-negative bacteria have an outer membrane rich in phospholipids and LPS, which is lipopolysaccharide. This outer phospholipid layer acts as a barrier to certain substances, including some antibiotics, whereas in gram-positive bacteria, it lacks this outer phospholipid membrane. Their cell wall is thick and mainly composed of peptidoglycan. Complement lysis. For gram-negative bacteria, it is more susceptible to complement mediated lysis because complement protein can access the outer membrane and form membrane attack complexes, which is MAC, MSC, causing bacterial cell rupture. For gram-positive bacteria, less susceptible to complement lysis due to thick peptidoglycan layer that prevents access to the cell membrane. Lysozymes, enzymes found in macrophages and other immune cells that degrade peptidoglycan. For gram-positive bacteria, it is more sensitive to lysozyme because of the thick peptidoglycan layer. For gram-negative bacteria, it is less sensitive since the outer membrane restricts enzyme access to peptidoglycan. Peptidoglycan layer for gram-positive have a thick peptidoglycan layer which provides rigidity and shape. For gram-negative have a much thinner peptidoglycan layer located between the inner cytoplasmic membrane and outer membrane in the periplasmic space. Teichoic acid. It is present only in gram-positive bacteria. Teichoic acids are polymers in the peptidoglycan that contribute to cell wall maintenance, regulation of cell growth, and antigenic specificity. LPS, lipopolysaccharide, it is present only in gram-negative bacteria. LPS is a major component of the outer membrane and act as an endotoxin contributing to the immune response and pathogenicity. Sensitive to beta-lactam antibiotics. For gram-positive bacteria, generally more sensitive to beta-lactam because penicillin targets peptidoglycan synthesis and gram-positive bacteria have abundant peptidoglycan exposed. For gram-negative bacteria, often less sensitive because the outer membrane can block penicillin access. Also, many have beta-lactamases that degrade these antibiotics. Spore formation for gram-positive bacteria, some genera like Bacillus and Clostridium form endospores for survival under harsh conditions. For gram-negative bacteria, they generally do not form spores. Plasma membrane. Phospholipid bilayer. The plasma membrane is a phospholipid bilayer composed of hydrophilic, which is water attracting, heads facing outwards, and hydrophobic, water repelling, tails facing inward. It is embedded within this bilayer are proteins that serves various functional roles. Selective permeability. The membrane controls which substances enter and exit the bacterial cell, allowing passage of nutrient, waste product, and signaling molecules while blocking harmful substances. Site of metabolic activities. It hosts crucial metabolic processes such as cellular respiration, photosynthesis in some bacteria, and synthesis of lipid and cell wall components. So this diagram is bacterial plasma membrane structure. This lipid bilayer, the basic framework of the plasma membrane is phospholipid bilayer. Phospholipids have hydrophilic, which is water attracting, heads facing outwards, and hydrophobic, water repelling, facing inward, forming a semi-permeable barrier. This bilayer provides membrane fluidity and barrier function, separating the internal cytoplasm from the external environment. Proteins are embedded or attached to the phospholipid bilayer and are critical for numerous functions. For example, protein channels and carrier proteins. This carbohydrate chain attached to proteins and lipids on the outer surface of the plasma membrane, forming glycoproteins and glycolipids. These carbohydrate chain play key roles in cell recognition, adhesion, and interaction with the environment or other cells. Other components, the membrane contain enzymes involved in respiration and other metabolic processes. It acts as a site for energy generation since bacteria lack mitochondria. Inside the cell, the plasma membrane lies in the cytoplasm, a gel-like substance where various cellular processes occur. Outside the cell, the plasma membrane is the cell wall, which provides additional shape and protection. This protein channel embedded in the phospholipid bilayer are protein channels, or you can call transport protein. This protein facilitates the selective transport of ions and molecules across the membrane, including nutrient uptake and waste removal. They enable passive or active transport, helping the cell maintain homeostasis. Cytoplasm. As I mentioned earlier, the cytoplasm is a gel-like semi-fluid substance enclosed within the plasma membrane. It is the medium that fills the interior of the bacterial cells. Contains enzymes, nutrients, ribosomes. Enzymes is nothing but catalyze various biochemical reactions essential for bacterial metabolism. Nutrients, organic and inorganic molecules necessary for energy production and growth. Ribosomes, tiny structures scattered throughout the cytoplasm where protein synthesis occurs. Other cellular molecules and components involved in the cell's biochemical activities. Sites of biochemical reaction, it acts as the site of many metabolic processes including protein synthesis, glycolysis, DNA replication. It provides a medium for the movement of molecules that are vital for cell function. Nucleoid. The nucleoid is an irregularly shaped region inside the bacterial cytoplasm that contains the circular DNA of the bacterium. Unlike eukaryotic cell, the nucleoid lacks a nuclear membrane. Thus, the DNA is not enclosed within a separate nucleus, and it acts as the genetic control center of the cell. Bacterial chromosome. This chromosome in bacteria is typically a single circular DNA molecule that contains most of the genetic information necessary for the cell's function and reproduction. This DNA is supercoiled and compacted to fit inside the limited space within the nucleoid. The chromosome contains genes essential for cellular metabolism, growth, and replication. Plasmid Plasmids are small circular independent DNA molecules found in many bacteria in addition to the main chromosome. They carry non-essential genes that can confer advantageous traits such as antibiotic resistance, toxin production, or the ability to metabolize unusual substances. Plasmid can replicate independently and can be transferred between bacteria through processes like conjugation. The nucleoid is situated within the cytoplasm without a surrounding membrane. So the diagram visually emphasizes the compact organization of DNA within the nucleoid and distinction between chromosomes and plasmids. Ribosomes in bacterial cells. Bacterial ribosomes are called 70S ribosomes, which are smaller than eukaryotic ribosomes, which are 80S. The S in 70S stands for Swedberg's unit, a measure of sedimentation rate during centrifugation that reflects size and shape. The 70S ribosome is composed of two subunits, the 50S large subunit, the 30S small subunit. Site of protein synthesis. Ribosomes are the site of protein synthesis in bacterial cells. They translate messenger RNA into polypeptide change by linking amino acid in the other specified by the messenger RNA. Protein produced are essential for various cellular functions, including structures, enzyme activity, and regulation. Prokaryotic ribosomes versus eukaryotic ribosomes. Prokaryotic ribosomes, which is 70S, comprised of two subunits, 50S large subunit and the 30S small subunit. It is smaller in size compared to eukaryotic ribosomes and found freely floating in the cytoplasm, whereas eukaryotic ribosomes, which is 80S, comprised of two subunits, the large 60S subunit and the small 40S subunit, larger and structurally more complex than prokaryotic ribosomes, found either free in the cytoplasm or attached to the rough endoplasmic reticulum. Both ribosomes synthesize protein by translating messenger RNA into amino acid change. Despite differences in size, the fundamental role in protein synthesis is conserved. So the diagrams typically depict the two subunits of each ribosomes with size proportions relative to each other. The prokaryotic ribosomes shows a simpler structure reflecting its smaller size and fewer protein RNAs. The eukaryotic ribosomes illustrate increased complexity and large subunit sizes. Mesosomes. Mesosomes are infoldings or invaginations of the plasma membrane in prokaryotic cells. They appear as folded or tubular structures extending into the cytoplasm. Mesosomes are analogues of the mitochondria present in eukaryotics. It thought to be analogues of mitochondria in eukaryotic cells and serves as the main site for respiratory enzymes and processes such as cellular respiration and energy production. It plays a role in DNA replication and distribution during bacterial cell division. So, diagrammatic representation of mesosome. Plasmid, again small circular double-stranded DNA molecule distinct from the bacterial chromosomes. It carries non-essential genes that can confer advantages such as antibiotic resistance, and it can replicate independently and be transferred between bacteria via conjugation. Mesosome invaginations or folds of the plasma membrane into the cytoplasm. This serves as a site for respiratory enzymes and energy production, aid in DNA replication and segregation during cell division, increase the surface area of plasma membrane, facilitating metabolic activities. Capsule, which is a thick gelatinous outer surrounding layer of the cell wall, provides protection against desiccation and phagocytosis by host immune cells. Capsules helps bacteria adhere to surfaces and evade host defenses. Then cell wall. Cell wall is a rigid structure made primarily of peptidoglycan layer. It provides shape and mechanical support, protects the bacterial cell against osmotic pressure, differs between gram-positive, which have thick peptidoglycan layer, and gram-negative, which has thin peptidoglycan outer layer of bacteria. Flagella, long whip-like appendages used for motility, can be singular or multiple arranged differently. Ribosomes, small granule structures, 70S in bacteria, composed of 50S and 30S subunits, sites of protein synthesis freely distributed in the cytoplasm. Nuclear material, which is nucleoid, region in cytoplasm containing bacterial chromosome DNA. DNA is circular and lacks a nuclear membrane, and nuclear material contains genetic function in cell processes. Inclusion bodies. Inclusion bodies are intracytoplasmic granules or aggregates found in the cytoplasm. They serve as storage reserves for essential nutrients, energy sources, or other substances. Functions such as storage of carbon sources such as glycogen or poly-beta hydrobutyric acid, which is PHB, storage of inorganic substances like phosphate, volatility on metachromatic granules, reservoir of energy useful for the cell during adverse condition, and helps in reducing osmotic pressure by storing substances internally, preventing cell lysis in hypotonic environments. Examples of inclusion bodies: metachromatin or volatilin granules, starch inclusions, lipid inclusions, poly-beta hydrobutyric acid (PHB). This metachromatin or volatilin granules store inorganic phosphate, starch inclusion store polysaccharide, lipid inclusion stores storage of lipid compounds, etc. The following diagram, this slide generally shows the bacterial cytoplasm with visible inclusion bodies depict as distinct granules or clusters. These granules or clusters. The structure appears as dense spot or particles within the bacterial cytoplasm. Capsule and slime layer. The capsule is a well-organized, dense, and firmly attached outer layer surrounding the cell wall. It is composed mainly of polysaccharides, and it provides protection against desiccation and phagocytosis by host immune cell. It enhances adhesion to surfaces and host tissues, facilitating colonization and infection. It contributes to bacterial virulence by helping evade host immune responses. Capsules are often visible under a microscope with special stain, that is capsule staining. The following diagram typically shows the bacterial cell structure with a thick capsule layer. The capsule is usually drawn as a clear or shaded halo around the outermost part of the bacterial cell. It is labeled as capsule to differentiate it from other layers such as the slime layer, cell wall, and plasma membrane. Pili and fimbriae. Pili and fimbriae are hair-like structures located on the bacterial surfaces. They are typically shorter and thinner. Functions of fimbriae primarily involved in attachment to surfaces, host cell, and other bacteria. It helps in colonization and formation of biofilms important for bacterial pathogenicity. Pili play a crucial role in bacterial conjugation, which is a process of DNA transfer between bacterial cell, and it also assists in adhesion and sometimes motility. Flagella are longer whip-like structure. They enable bacteria to swim through liquid environments. Types of flagella are monotrichous, lophotrichous, amphitrichous, peritrichous. Monotrichous is single flagellum at one pole. Lophotrichous is cluster of flagella at one or both the poles. Amphitrichous is flagella at both poles, and peritrichous is flagella distributed all around the cell surface. The diagram typically illustrates a bacterial cell with pili, fimbriae, and flagella. Fimbriae and pili appears as short, fine hair-like projections all over or at specific points on the surface. Flagella are shown as long and thicker whip-like appendages extending from one or more positions. The diagram may also highlight the different flagellar arrangement which is mentioned above. It visually differentiates the structures to show distinct roles of pili, fimbriae for attachment and conjugation and locomotion. Arrangement of bacterial flagellars. Types of bacterial flagellum arrangement. I'll explain one by one. Monotrichous, a single flagellum located at one end pole of the bacteria, which has only one flagella. Example, Vibrio or cholera. Amphitrichous, a single flagellum or tuft of flagella at both the ends poles of the bacterium. This arrangement allows movement in both directions. For example, alkali genes. Petritrichous flagella distributed all over entire surface of the bacterial cell. This arrangement provides powerful motility. For example, E. coli and Salmonella. Lophotrichous, a cluster or tuft of flagella at one or both the ends of the cell, example Pseudomonas species. Atrichous bacteria lack flagella completely. There's no flagella. It means no motility through flagellar movement. These bacteria rely on other mechanisms for movement or survival. Cephalotrichous. It is less commonly used term that refers to a cluster or tuft of flagella located at one pole of end of the bacterial cell. It is essentially synonyms with lophotrichous flagellar arrangement. This arrangement allows bacterium to have enhanced motility by rotating multiple flagella at the pole. It is a detailed structure of bacterial flagellum diagram. We'll start with filament, the long whip-like external part of the flagellum. It is composed of flagellin protein, function as a propeller for bacterial motility. This hook a curved flexible segment connecting the filament of the basal body and act as a universal joint for transmitting motor to the filament. Basal body. Here, L ring is there. It is located in the outer membrane of gram-negative bacteria. It stabilizes the flagellum by anchoring it to the outer membrane. P ring, which is peptidoglycan ring, situated in the peptidoglycan layer cell wall, which provides rigidity and support as the flagellum passes through the cell wall. S ring, it is a part of the cytoplasmic membrane or just beneath it. It is a part of basal body structure that helps anchor the flagellum. H ring, sometimes noted in diagram, an additional ring associated with outer membrane stabilization. This outer membrane, the outermost layer of gram-negative bacteria containing lipopolysaccharides, the L link is enabled here. Hello,
Please tell, madam.
my PPT is visible.
Yeah, yeah. But you are not visible.
I, I'm not visible, sir.
No, no, you are not visible.
Is it visible?
Yes, right now.
Where I was outer membrane layer.
Again, camera is off.
Hello.
Again, camera is off, I think.
Is
Now.
it visible now, sir?
Yes, yes, yes.
Okay, so outer membrane, the outermost layer of gram-negative bacteria containing lipopolysaccharide, peptidoglycan layer, the rigid cell wall providing structural support, periplasmic space, the space between the outer membrane and the plasma membrane filled with gel-like material, plasma membrane, the innermost cell membrane controlling molecule entry and exit, mod proteins. Here there is two mod proteins, mod A and mod B. It is located in the plasma membrane, forms the stator of the flagellar motor, allow proton flow, that is, which generates torque to rotate the flagellum. C ring, it is located inside the cytoplasm, attached to the basal body, functions in rotation, direction switching, and motor protein attachment. Endospores. Endospores are dormant, resistant structures formed inside some bacterial cells and which is under harsh condition. They allow bacteria to survive in harsh conditions environment that would be normally kills the cell, for example, Bacillus and Clostridium. Types of endospores. Spherical center, oval subterminal, spherical subterminal, and oval terminal. So we'll start with spherical center. It is location formed in the center of the bacterial cell, shape round or spherical. It affect the bacterial cell shape, remain mostly unchanged. Example, Bacillus subtilis often has spherical central endospores. Oval central location is oval central within the cell. Shape is oval or elongated, causes slight distortion or swelling of the cell. This variation often depends on bacterial species. Oval subterminal, it is location formed near the end. It is distorted near the end but not at the pole of the bacterium. Shape is again oval, causes slight swelling. Cell shape may be slightly distorted. Again, spherical subterminal location is also subterminal. It may cause slight swelling of the cell, less common than the oval subterminal. This is a diagrammatic representation of endospore structure. Core, the innermost part of the endospore, it contains bacterial DNA, ribosomes, and essential enzymes. Metabolically inactive but maintains the genetic material and has lower water content, which contributes to resistance. Inner membrane surrounds the core, act as a permeable membrane, protects the core contents and controls molecular transport. Germ cell wall, a thin layer, which you can see here, a thin layer surrounding the inner membrane, becomes the cell wall of the new vegetative cell upon germination, maintains the shape and mechanical strength of the spore core. Cortex, it is slightly thick layer made primarily of peptidoglycan, lies between the germ cell and outer membrane, plays a crucial role in maintaining spore dormancy and dehydration, protects the core from the heat and chemicals. Outer membrane lies outside the core. It lies outside the cortex, a lipid bilayer that further protects the spore, less understood but contributes to maintaining the spore's integrity and act as a permeable layer. Coat, a multi-layered proteinaceous layer outside the outer membrane, which provides chemical and enzyme resistance, protects against destructive enzymes and toxin chemicals. It gives mechanical strength to the endospores. This is a diagrammatic representation of endospore formation cycle. We'll start with vegetative cells. The actively growing metabolically functioning bacterial cell, it is under unfavorable condition like nutrient depletion, it initiates sporulation. Force spore, the initial small compartment that form inside the vegetative cell after asymmetric cell division, it will develop into the mature endospore. It contain a copy of the bacterial DNA. Mother cell, the larger compartment surrounding the forespore, supports and nurtures the development of the forespore into an endospores, ultimately lyses to release the mature endospores. Here is the DNA replication. The bacterial DNA is duplicated in preparation for cell division. Asymmetric cell division, the cell divides unequally, forming a small forespore and a larger forespore, which is called as mother cell. And then engulfment, the mother cell membrane engulfs the forespore, surrounding it with a second membrane. Cortex formation, a thick layer of peptidoglycan cortex forms between the two membranes of the forespore. Maturation and dehydration. The spore mature, becomes dehydrated, and develops a resistance to heat, radiation, and chemicals. Lysis of mother cells. Here, the mother cell finally break down into lysis, releasing the mature endospore to the environment. Lastly, germination. Again, when the condition improve, the endospore germinates back into a vegetative cell, reassuming growth and metabolism. Questions. 3-mark questions. Fimbre and draw a labeled diagram of bacterial cell. For fimbre, you can explain what fimbre are and their role in bacterial cells, particularly in attachment and colonization. Draw a label diagram of bacterial cells. Here you can create a neat label diagram illustrating the major components of a bacterial cell, such as cell wall, plasma membrane, nucleoid, ribosomes, flagella, pili, capsules, etc. For 5-mark questions, draw a neat label diagram of bacterial cell and explain. Here you can draw a detailed bacterial cell diagram with all major structures clearly labeled and provide an explanation of the function of each part. Bacterial flagella, explain the structure, function, and types of flagella in bacteria, including their role in motility. Bacterial spores, here you can describe what is bacterial spores, for example, endospores, their formation, structures, types, and significance. Bacterial cell wall, here you can explain the composition, structure, and differences between gram positive and gram negative bacterial cell walls. Thank you.
Thank you, Dr. Namira Khan. It was very nice presentation. Alhamdulillah,
Thank you,
you have
sir.
covered fully about cell wall and the cell structure. Thank you.
Yeah,
Thank you so much, madam.
thank you, sir.
Thank you. So, shall we wind up, madam?
Yes, sir, we can wind
Thank
up.
you. Thank you.
Thank you. Mm.